Mast Cell Activation Syndrome. What is mast cell activation syndrome in terms of a biotoxin illness, in terms of a mycotoxin illness, in terms of Lyme disease and co-infections, and anyone who has immunocompromisation? What does mast cell activation actually mean? What does MCAS actually mean? What is the key mechanism? That's what the video is all about. So essentially, so every pathogen, so every living pathogen emits what's called a pathogen-associated molecular pattern. So on the outer cell, or on the outer layer, or just on the outer coating, whatever you'd like to call it, because pathogens have many different outer layers, variants. They have many variants of outer layers. For example, the outer layer of a virus is completely different from the outer layer of a yeast, and the outer layer of a bacteria is completely different from the outer layer of an archaea. So let's just call it outer layer. Whatever pathogen is in the body, and a mycotoxin, for example, a mycotoxin also emits an outer layer. So whatever outer layer is in the body is going to emit a pathogen-associated molecular pattern. Okay, and the body, our human body, our cells recognize these pathogen-associated molecular patterns. They're known as PAMPs, 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 pathogen-associated molecular patterns. Now, we have pattern recognition receptors. So our immune cells have pattern recognition receptors. And depending on the type of immune cell, whether it's from the innate immunity, which deals with mycotoxins, or the adaptive immunity, which majority of the time deals with viruses, these immune cells have different versions of pattern recognition receptors that pick up and basically identify different pathogens via the pathogen-associated molecular patterns. So on the pathogen, you have the pathogen-associated molecular pattern, and on the human immune cell, you have pattern recognition receptors, depending on which type of human immune cell, whether it's innate or adaptive. And you can either categorize innate and adaptive even further into subcategories. They'll have different pattern recognition receptors. Okay, so that's been decided and that's been established. So when a pattern recognition receptor from the innate immune system, which deals with a mycotoxin, picks up a mycotoxin-specific pathogen-associated molecular pattern, this is known as antigen signaling, okay? So this sets off a whole bunch of inflammatory cascades like the complement factor, the complement immune factor, which is why in mold illness and CIRS and biotoxin illness, we often test for C4A. C4A is a blood marker that is basically an end product of the complement immune factor, the complement immune system factor, that signaling cascade. And this is all downstream to the pattern recognition receptor picking up on the pathogen-associated molecular pattern of the human and of the mycotoxins. You can see how it's all downstream. It's a big, strong signaling cascade. Okay, so when antigen signaling does occur, when antigen signaling occurs, you have a lot of inflammation. So you have a lot of inflammatory cytokine release, you have a lot of inflammatory interleukin release. The immune cells are going crazy, they're trying to differentiate, they're trying to get a hold of the infection, they're trying to reduce the antigen signaling, because essentially their pattern recognition receptors are ringing the alarm bell. So everyone's panicking, okay? So essentially, when this happens, and when this happens for a really long time, so when this happens for an extremely chronic and an extremely long time, chronic inflammation, this constant signaling from the pattern recognition receptor due to the pathogen-associated molecular pattern ends up in mast cell activation syndrome, okay? And it's a vicious cycle because the more your mast cells activate, the easier down the line they activate further, okay? So when a mast cell, when it does activate and it releases granules and it does actually activate and it releases a lot of pro-inflammatory cytokines, pro-inflammatory interleukins, the whole mast cell ecosystem, the whole mast cell organization system is even more likely to activate with an even smaller trigger later down the line. So you end up in an extremely vicious cycle when it comes to mast cell activation syndrome. When the mast cell degranulates, when a mast cell degranulates due to chronic inflammation, every other mast cell further down the line that degranulates is going to degranulate with a lesser trigger. So it's going to take even less to have even greater mast cell degranulation. So you end up in a very vicious cycle. That's what I'm trying to say. So I'm going to do a little slideshow here of some infographics, and these infographics are infographics I've made. I designed these infographics and they're on my X and they're on my Instagram. And these infographics basically give you a demonstration of pattern recognition receptors and pathogen-associated molecular patterns. So if you prefer an in-text explanation, if you prefer a visual and in-text explanation, then you can find these infographics on my X and my Instagram. So I'm going to place them here. Oh, and also I have a board. Yeah. So I actually, I drew all of this mechanism out onto a board, Zoe me as well. I'm always forgetting about my boards, even though I love them. So I drew this key mechanism out on my board as well. So I'm gonna move aside and I'm gonna put my board somewhere here. And this board is also the key mechanism of mast cell activation syndrome. Key mechanism of mast cell activation syndrome. Okay, so, actually, how can we interrupt this degranulation? How can we interrupt antigen signaling at the cellular level? How at the level of signaling, very, very, very subcellular, cellular level, cell membrane level, organelle level, cytokine interleukin level. How can we actually interrupt this whole process and therefore stop the sensitivity of the mast cells and therefore reduce the trigger points and therefore reduce their granulation? Well, actually, peptides, also phospholipids, also binders. Binders are actually very important. I know I don't speak about binder therapy too often, just because I think it's so simple. I just think it's so simple. You just go online, order a binder, take it away from food, take it 45 minutes away from food before and after, 2 to 3 times a day. I just think binders is just so simple, and I actually don't think anyone needs to be coached on how to do binder therapy or have a consultation on binder therapy. It's just so simple, you just take a binder. Anyway, so binders, they are important in this instance. So binders actually, and there is a use for binders, by the way. Just because something is simple and just because I categorize binders as simple doesn't mean that there's no use. Binders definitely need to be used in immunocompromisation. It's just very simple, so I'm not going to touch upon it. you can basically describe binder therapy in 1 or 2 sentences. Anyway, so binder therapy in this instance, it's heavily needed because you do need to physically, drag and remove the antigen out of the body. You do need to drag the antigen, you do need to, drag the pathogen that has the pathogen-associated molecular pattern on its outer layer out of the body. So you do need to bind and remove the pathogen. So binders are all good here. So yay, binders. And then phospholipids as well. So phospholipids are really important. Obviously, mast cells have a phospholipid outer membrane. They do. Mast cells have a phospholipid outer membrane. And in this phospholipid outer membrane, that's how they degranulate. So they have a signaling molecule come, tell the mast cell, ooh, gotta go, fire's burning, time to degranulate. So we need to repair the outer phospholipid layer, the inner phospholipid layer, all of the organelle phospholipid layers of the mast cell in order to decrease its sensitivity and therefore stop the mast cell from degranulating. That's an understatement. My favorite one and only phospholipid product is the BodyBio PC. I'm gonna put it here. I love the BodyBio team. I've been working with the BodyBio team for, I think it's been nearly 2 years actually. 2 years in August. So I started working with the team in August 2023. Super amazing team. You can find all of my content on BodyBio, on phospholipid therapy, on lipid replacement therapy, all of my X and all of my Instagram as well. And we're going to be writing some co-articles about phospholipid therapy together soon, so it's going to be very interesting. But we need to replace the phospholipids in the mast cell in order to avoid degranulation. My third point, which I'm going to discuss, which is one of my key methods of actually reversing mast cell activation at the cellular level, is peptides. Why? Because peptides actually exist and signal at the cellular level. So that's what peptides actually do. So peptides, they don't exist at any other level. They're very small, well-organized, well-structured amino acids that signal at the cellular level. Peptides interact with G protein coupled receptor membrane proteins on the cell membrane of cells and signal. Just like hormones. Hormones signal like peptides. They have the same signaling power. The same signaling power, except hormones are made from cholesterol, they're steroidal-based, pregnenolone-based. Peptides are amino acids, okay? So, and actually we have much wider use in peptide therapy than we do in hormonal replacement therapy. There's only, a certain amount of hormones, right? You can't wake up one day and make a new hormone. Well, I don't know actually. It's got a good idea. Anyway, to my knowledge, you can't wake up one day and make a new hormone, but you can wake up and make a new amino acid sequence and help people. And that's something I want to get into. I want to get into actually making new amino acid chains. I'm very lucky. My community often send me very recent. I'm actually incredibly lucky. My community, we have a very good feedback loop, which, I'm so grateful for. I'm so lucky. And my community often send me very new amino acid chains, which are not yet on the bench. So we don't have these at the lab. And a goal of mine was to actually maybe get into the processing and development of these new amino acid chains so we can help more people. I think that's an extremely interesting venture, and that's something I want to do. So thank you so much for my community for that. So yeah, thank you. So as I was saying, we can use a whole bunch of peptidos, which is peptides in Japanese, peptidos. We can use a whole bunch of peptidos to actually signal to the immune cells, hold on, stop, no need to cause mast cell granulation, Signal to the thymic gland, hold on, stop, calm down. No need to overreact to this trigger. Signal to the brain, to the hypothalamus, to the pituitary. Signal to the mast cells. Signal, 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 signal. Cause a big signal. And basically tell all of the cells, all of those inflammatory cells to stop. So, you know, if you have a high load of inflammatory cytokines, a very high load of inflammatory interleukins, then we can inject you with exogenous, we can supplementing with amino acids. We can supplement you with exogenous peptides, and we can actually rebalance pro-inflammatory cytokines and anti-inflammatory cytokines, pro-inflammatory interleukins and anti-inflammatory interleukins, which is awesome. Yeah. So I hope my explanation made sense, because who knows, it makes sense in my head. You know, I hope it makes sense with you as well. If you have any questions, you can ask me below. And also I love references. I love reading. I love to research. Anyone who knows me, in the Zoe Me group chat, I'm always sending in like references like all the time. On a Saturday, on a Sunday, on a Monday, on a Tuesday, on a Wednesday, on a Thursday, on a Friday. I'm always sending in references because I just pick up on scientific papers. It doesn't take me too long. I can just read the title and automatically know and understand the kind of like key mechanism. So anyway, my main point is that I'm going to put all the references for the knowledge that I've tried my best to facilitate to you below in the YouTube description or below in the caption. So you'll find all of those. They're going to be hyperlinked. A lot of them are on PubMed as well, and a lot of them are open text, so you can fully read those. Of course, you know, if you want to work with me, say you have Mast Cell Activation Syndrome and you want to work with me, you think we'd be, you know, a good fit working together, I can definitely help with your Mast Cell Activation Syndrome. We can use a wide range of therapies. If you're in Dubai, we can do that face-to-face at Zoe Me with my team, with my amazing team. And if you're remote, then of course I can see you remote as well. I've seen hundreds of clients remote as well. So that's really interesting. So if you do want to work with me, then the links to work with me are below as well. There's my link in bio, and then I think I have my email there too. And then there's a direct link to book a discovery call with me. And if you want to keep up more with my research, then you can follow me on X, you can follow me on Instagram, you can subscribe to my YouTube channel, whatever you feel like doing, whatever resonates with you. I talk a lot about immunocompromisation and regenerative medicine, peptides, bioregulators as well. And I also post a little bit about fashion and, you know, growing as a person as well, which is also all a part of science too. As you get healthier, so, you know, I do notice this in my client community, my client population, and my friends as well, everyone who I love and care about. As you get healthier, you get less rigid, okay? You get less rigid, you get more spontaneity, you get more spontaneous, okay? So you get more spontaneous as you get healthier. And I see that in my clients too. And I hope to reflect that as well in who I am, of course. And yeah, so I just want to talk about, you know, closing thoughts. So thank you so much for listening. And before you go, I just want to talk about my look. So, okay. So let's break it down. So I have on a top that is like a spider. So it's a really interesting, like it's like a mesh. Here, yeah, you can see. So it's like a mesh top, and it's like a spider. I think the inspiration might have been Spider-Man. I don't know. Spider-Man is not my favorite superhero. No way. I don't like Peter Parker. he is, I could beat Peter Parker up, like, no. I like Batman. I don't think I could pull a fast one on Batman. Yeah, I like Batman. So yeah, this is Spider Top. I've done pigtails with bows, so I have some really cute bows. I love bows. And then as well, I don't like to leave the ends of my hair open, you know, like when you just have like a little plastic clear scrunchie. No, I don't like that. I don't think it looks done. It doesn't make you look done. So what I actually have is like, I buy multiple different ones, so you can see. So I buy these and I always put them on the ends, and you just look so much more put together, especially when you do like braids. When you do messy braids, you go into the gym or something, you put these little ends in, done, looks so together. And then if we talk about my makeup, which I love to do, I don't have any foundation on. So I don't have any concealer, I don't have any BB cream, this is just moisturizer. I look after my skin. I am extremely territorial about my skin. I look after my skin because I haven't had the best skin journey. I've had a really bad journey with my skin from a very young age. So I'm extremely territorial about my skin. So I really look after my skin. So I'm quite lucky I have a very clear complexion without any makeup. I would probably give most of that credit to the sauna. I do go to the sauna 5 times a week and I really sweat and I take binders and everything in the sauna. And also my gut health. My gut health is pretty good too. And when you have good gut health, good muscle tone, you have very good skin. Oh, and also, also I should say, I'm really big into regenerative aesthetics. So I do get a lot of salmon polynucleotides. I do get a lot of exosomes injected into my face, and I get a lot of facials as well. So I think that also plays a large part. On my lips, I have a Sephora brown, no, Kiko. I have a Kiko brown liner, and then I have a Kiko like shimmery purple gloss. It's really, really nice. I spoke about in a video a while ago about how I was trying out the new brown gloss liner trend, and I love it. But it only goes with certain outfits. Like, it doesn't go with pink, it goes with these very dark outfits I like to wear sometimes. Okay. And on my eyes, so I just have a black eyeliner. So I just have a black liquid eyeliner as a cat eye, medium cat eye, not short, not long, medium. And then I went into my upper waterline with a black kohl and painted my waterline. I didn't want to wear mascara today. I just wanted to leave my natural lashes, so I just curled my natural lashes. It goes. I think it looks more kawaii and cute when you don't have any mascara on, but you still have the black liner. And then I put white kohl in my lower lash line, just to make my eyes pop. And then my earrings, they are from a store online. They're from a gothic store that I go to online. Try and get you a close-up. So my earrings are just from a gothic online store. I have like different moods, and one of them is emo goth, and yeah, I love this. But like I said, if you would love to work with me just like I would love to work with you, then you can check out my details in the description below. You can also find all the references too. Thank you so much for listening. Thank you so much for watching. It's my community which, you know, really makes the videos. It's my community that gives me these ideas. So I'm very grateful for you. I'm very grateful for every single one of you. So thank you so much, and I will see you very soon.